sound,
Sound.
speed, rolling,
Action.
action. So welcome to Essentials of Computational Chemistry from OpenEye Cadence Molecular Sciences. Today, part 2, structure-based design, and I will be your host for this journey. My name's Paul Hawkins. So if you've seen part 1, you will already know that what we're aiming for here is a relatively shallow but wide look at some significant problems and solutions in structure-based design. So there's a great deal of material in the supplementary, some hidden slides, so when you finish listening to the presentation, please download that and burrow further into any topics that seem to interest you. Before I get into the technicalities though, I'd like to ground what we're doing in what we're doing, which is of course the discovery of human medicine. We want to get new medications in the hands of patients who need them as quickly and as cost effectively as possible. So what I show here is an 8-stage outline of the drug discovery process, stages 1 to 4, discovery, and then 4 to 8, development. As you can see simply by inspection, this is a long process, it's a very expensive process, and it's a very risky process. About 10,000 molecules need to go in at the beginning of this process to have a high probability of one emerging at the other end successfully. I can't think of another business where you'd be in business if 99.99% of all your ideas failed. So what we do here in the Essential series, what we do at OpenEye as software developers, is develop and test computational tools mainly to assist with the first 4 stages of drug discovery. So I will use this outline of drug discovery, a nice, neat chevron-based process workflow. But I remind you that anything involving science, and particularly the science of human biology, is a lot more complex than that. I'm borrowing here a picture from Rob Morcher-Smith at AstraZeneca. The goal is to get the drug into the hands of the patient, but unfortunately, the process is not linear, a lot of sideways and backward steps, as many as there are forward. But I'll continue to stick with the rather neater chevron outline. It's much easier to follow. So I'm going to look at 2 stages of drug discovery today. Firstly, the compound screening stage, where we're going to use computational methods to find new molecules that have interesting biology on our target protein. And then I'm going to look at lead optimization, which is trying to manipulate molecular structure to improve a series of drug-relevant properties. So I'm going to look today at structure-based methods specifically, but obviously, as you may know from part 1, there are multiple ways to attack any problem in this area. And I'm going to compare these problems on 3 different axes. The y-axis here on speed, the z-axis on accuracy, and the y-axis for novelty. As you'll see later, with the rise in the size of available molecular space, speed becomes more and more important. An enormous draw for structure-based methods, and particularly the topic for today, docking, is novelty. Ligand-based methods, be they 2D or 3D, will inherently introduce some bias, some similarity to known ligands. And a great appeal for structure-based methods, docking included, is that they are unbiased or naive to molecular structure and therefore can find new and interesting molecular structures. And again, that supports the intellectual property discovery part for commercial drug discovery. So, what is this magic docking? Well, it's a pretty simple idea. We're going to take a set of molecules we'd like to screen, and I'll show you how to get conformations for those, and then we're going to have one or more conformations of the protein we're going to be looking at, and then, through the magic of docking, success. And I'm going to define what success looks like in the context of early stage drug discovery in the middle section of the talk. So, very briefly then, we need to get molecules ready to be docked, and generally speaking, docking tools are going to require one or more 3D conformations, arrangements of atoms in space, for every molecule they'd like to dock. Unfortunately for me, but perhaps fortunately for you, I don't have time to go into conformation sampling today, so if you'd like to know more about that, please see Essentials Part 1. So, with the molecule conformations out of the way, the experimental conformation of the protein. I'm going to stick to experimental conformations today. There are many theoretical methods to obtain atomic coordinates for proteins, but I'm not going to touch that. I'm going to stick explicitly to experimental methods. There are 3 key experimental methods most used to get atomic coordinates for proteins: X-ray crystallography, NMR, and cryo-electron or 3D electron microscopy. And I will look today at the first, X-ray crystallography, and the third. We won't be covering NMR today. Crystallographic approaches have been in use for many decades and have been used to get very, very fine resolution of structures. Cryo-electron microscopy has become much more popular in the last decade or so with what's called the resolution revolution. And as you'll see, these structures are now becoming competitive in terms of their precision with structures from crystallography. But no matter what the particular structural biologist's source of their data is, they have the same problem. And that is, they have to take data, in this case this is electron density data from crystallography, and they have to take atoms, and they have, using a combination of sweat, skill, and software, put the atoms into the density to make a model. So the atomic coordinates that you're consuming when you're doing docking or other structure-based design aren't the data, they're a model of the data. And so obviously a question that comes to everyone's mind is, how can I discriminate a good model from a bad? I have good news. Never fear, the WWPDB is here. The Worldwide Protein Data Bank produces a structure validation report for every single structure in their database, again, no matter what the technique used to produce the coordinates was. And we'll look at the validation report first for crystallographic structures. And it contains 5 different criteria, the first and last of which relate to the coordinates and their match to the crystallographic density. So the R3 relates to overall global model quality, and the RSRZ relates to local model quality matched to side chains, and so on. The middle 3 relate to the coordinates themselves, so looking for interatomic clashes, which are unphysical, bad, looking for Ramachandran outliers for protein backbones, and also for side chain outliers. So the structure here is for chicken lysozyme at about 2.1 angstrom resolution, and that's the median resolution for structures in the PDB and has been for about 2 decades. So as we look at this structure, we can see that the metrics get better to the right. This is a very poor structure. It's a very poor match to its local, its global models density. The clash scores are very high. It has a lot of backbone orientation outliers and a good number of side chain outliers also. This structure, on the other hand, is also from chicken lysozyme at a slightly worse nominal resolution, but it's a much better model. Overall, it's a far better fit to the global density, it's a better fit locally, and has far fewer problems with the atomic coordinates. So here we're seeing that the slightly worse nominal resolution structure is actually a better model. So resolution can tell you how good the model could be, but it doesn't tell you how good the model is. Another thing that resolution is quite awkward to do is to look at particular local model quality metrics. So I'll give you an example here. 2 ligands in 2 different structures, one here on the left, a very good fit to density, as you can see by eye. The atom coordinates fit nicely into the density. In the second example, a lot of the ligand coordinates atoms are not covered by density, and actually the conformation of the molecule here turns out to be wrong. The WWPDB has data for this also. They provide both the RSCC, the real space correlation coefficient, and the real space R factor. RSCC should be higher, RSR should be lower, so you can see here good RSCC, it's high, and low RSR. For the other structure, much lower RSCC, worse, and much higher RSR, worse. So again, resolution here not telling you a lot about local quality, but there are other metrics that you can use to help with that. So to help us get a good handle on structural model quality from crystallography, here at OpenEye we developed a set of criteria we call the Iridium criteria. 5 of these criteria relate to global quality, including something called the Diffraction Coordinate Precision Index, DPI. This is a little bit like the uncertainty in a measured log P, or solubility. It tells you something about the inherent uncertainty in the atom positions in the model, lower uncertainty being better. And then 5 criteria for looking at the active site, the binding site particularly. These criteria can be used to classify structures into 3 different classifications. The highly trustworthy structures are pretty rare, about 6 or 7% of the PDB currently. The great majority of structures are moderately trustworthy. About 1 in 6 or 7 are not to be used for modeling except in extreme circumstances. And the classification can be used to identify a particular issue, and then that can either be fixed manually or automatically if you're lucky, or if the problem can't be amended, then discard the structure and move on to another. Speaking of moving on, the other approach to structural models at the atomic level is cryo-electron microscopy, and that's become, again, massively more popular for the last 10 years or so, looking at a great deal of different types of protein that are hard to address with crystallography. Membrane-bound proteins, hard to crystallize. Large complexes, also hard to crystallize. And crystallography struggles to characterize proteins that have a lot of dynamic motion. These three can all be much better addressed through Cryo-EM. So as with X-ray crystallography, the WWPDB provides a quality metric guide here. It contains only 3 measures, the 3 that relate to the atomic coordinates. This is a structure of beta-galactosidase at about 2 angstrom resolution, just the same as the great majority of structures in the PDB, although about 85% of PDB structures are from crystallography, not Cryo-EM. So Cryo-EM here really showing very good resolution. There are a set of different criteria to match atomic coordinates to the Cryo-EM data. The community is still converging on the best criterion. I expect the WWPDB to add a global data fit quality measure in the next few months. But no matter where the coordinates come from, how the model was arrived at, it's important to think carefully about what application you want to put a model to. You're never going to get reliable predictions from an unreliable structural model, and you're never going to get high precision predictions from a low precision model. So think carefully about matching the model that you use in your structure-based design to the kind of results, the quality of results that you'd like to get out. More bad news, no matter where the structure comes from, X-ray crystallography, Cryo-EM, internal work, public databases, the structure, the model, won't be ready for modeling. It needs to be prepared. So here I've got an example of cyclin K. The asymmetric unit from crystallography is a dimer of dimers. So there are 4 protein strands in the asymmetric unit. 2 biological units, the ones that give the activity we see in the assay. Only one of those 2 binds a ligand, so we have 4 chains but only 2 ligands. There are missing pieces in the structure. Lots of obvious issues with this protein before it can be used for modeling, including the lack of hydrogens. More or less, no matter how you solve your structure, hydrogens will not be located experimentally. That can only be done with neutron diffraction. So you're going to have add your hydrogens computationally. I'll talk more about that in just a second. There are some also more subtle problems with protein structures. For example, here you can see this lysine residue isn't really defined with density. It's a very tiny piece defining one atom. Mostly it's defined only to the beta carbon. Residues and ligands in the binding site and cofactors can have alternate locations, partial occupancy, so that has to be taken into account. As I've showed you already, there can well be interatomic clashes that should be resolved before modeling, and the ever difficult problem of where water is in the binding site. So all these things together mean that the structures straight from the PDB aren't going to be ready for modeling. They need to be prepared. Great many different steps involved here. The first step will be to identify issues with the protein. The Iridium classification can help with this. And you can either choose to resolve the problem, again manually or automatically, or discard a candidate structure, move on to another. Generate the biological unit, identify the binding site, identify if there are impinging residues from other protein chains, so-called packing residues. Then we can add missing loops and tails, add hydrogens, again computationally, which may involve a force field refinement, and then we have something approaching a prepared receptor ready for modeling. One last step that may or may not be required is the addition of partial charges. That's necessary only for some docking tools. In contrast, if we want to do molecular dynamics, we'll have to move to a higher level of preparation, a full physics-ready model. If you want to learn more about this and other aspects of molecular dynamics for lead optimization, please join me for part 3. So, now we have the requirements, we have the molecules we'd like to process, and we have the protein conformation. So now we're going to do some docking, and we're going to look to use docking for 3 different but related problem types. First, screening, separating a database of molecules so the active molecules come to the top. Probably the most popular application of docking over the last 2 or 3 decades. Second, given a molecule is active, how does it arrange its atoms in the binding site? How does it interact with the protein? And can we use that information to design more potent analogs? And lastly, and most difficultly, ranking by binding affinity. So rank a database of molecules so the active molecules are in order of some estimation of their binding affinity. Very challenging problem. Not identical problems, but they're all overlapping. So to get into any of these 3 problems, docking is going to split the problem into 2 parts: posing and scoring. Firstly, placing the molecule in a binding site, understanding how it interacts with the protein. And there are lots of different algorithmic approaches to how docking programs approach this, and I'm going to divide them into sort of oppositional pairs. 4 different sets here, and I'll explain each one briefly in the next section. So for posing methods, a lot of approaches early on looked at the enormous possible pose space. If you have a large volume binding site and a flexible molecule, generating poses for the entire binding site can take a lot of compute time. So a lot of developers wanted to sample into this space without exploring it completely. And there are a couple of different approaches to sampling. One is to use a stochastic approach. Now, stochastic methods have the advantage they can search widely and in some cases in a guided fashion. But a disadvantage is that they're inherently irreproducible. The same input will give you a different output, which may need then multiple runs to see convergence, which increases compute time, but also, unfortunately, human time. The contrasting approach are deterministic methods, either looking to subsample in some sort of funnel fashion or a complete sampling of the space. Advantages here is you'll get the same output for the same input. That's how deterministic methods work. Disadvantage is that if the pose space is large, the compute time to explore that whole space can be too long. That's why a lot of developers have turned to a class called rigid docking, where we not only keep the protein rigid, as is almost always true, but the ligands also. So here we pre-sample conformations for the molecule, and the docking program doesn't have to change the conformation of any of its molecules. It simply places the molecule around the binding site, translating and rotating rigidly, scoring, eliminating low-scoring poses with a possible post-refinement and rescoring. This is how, for example, one of our rigid docking tools, OpenEye Fred, works. Most of these tools, all of these tools really are deterministic, same output for the same input. Pros here is that since we're not doing conformational sampling during docking, these tools can be fast, as you'll see later. A disadvantage is these tools place a great deal of stress on the conformation quality coming out of the conformation generator. If the conformation generator produces a suitable conformation, the docking program can find it. But if the conformation generator does not produce a suitable conformation, rigid docking will fail. So a lot of pressure on conformational sampling here, which is why a lot of developers use flexible methods. Here, one or a small number of conformations are fed in, and the docking program changes the conformation to better fit the binding pocket. So this is not done sort of a priori, but it's done with relevance to the binding site. We can use a sampling approach, stochastic methods, or we can use a funnel of deterministic searching, and this is the search funnel from the Glide tool from Schrödinger. There are a great many tools in the flexible docking area, again, some stochastic and some deterministic. These tools offer the probability of high pose accuracy because the pose is being generated in the binding site. It's relevant to the binding site environment. Disadvantages include lack of speed because, again, you're flexibly dealing with a molecule that may have a great deal of conformational space. So to take advantage of the speed of rigid methods but the accuracy of flexible methods, some developers have turned to what we call build-up methods. So they split the molecule into pieces, pose rigid pieces which have no conformational space, and then build up the flexible linking pieces in sequence. So there's an anchor fragment that's matched to features in the binding site, and then the molecule is grown step by step. And there are a number of approaches to this. These have the advantage that they can be pretty fast, as you'll see in a moment, but they place a great deal of stress on the docking engine's ability to place and score the anchor fragment correctly. If the anchor fragment's placed correctly, the probability of a good pose is high. If the anchor fragment is placed poorly, a build-up method cannot come back from that mistake. So it puts a lot of pressure on that early step. Now, all of these methods have this advantage that we consider traditional for docking, which is it's unbiased or naive. They're not biased by anything to do with ligand information, even though in most cases docking is going to be done into a crystal structure with a ligand, a small molecule, already in the structure. To take advantage of the information from that ligand, there are a class of docking tools which I'm going to term Ligand-Assisted. So these are going to use some piece of information from the ligand to increase the probability of a good pose or increase speed. So for example, we might use the volume occupied by a crystallographic ligand to reduce the pose volume in the binding site, reducing the calculation time. We may use match to the ligand in terms of either atom matching, shape matching, feature matching, to either guide the ligand, the molecule, into the binding site, or to even assist with scoring. So these ligand-assisted methods can take a variety of forms. They can be flexible or rigid. They can use the ligand information purely for posing, or they can use it for both posing and scoring. purely from first principles, using both ligand and structure information ought to improve the quality of results, and by reducing the pose volume, these tools will generally be faster. A disadvantage is we now lack the unbiased nature of docking. We've biased the posing towards the volume occupied by the ligand, and we may be biasing the chemical structure of new molecules toward those already existing in the chemical binding site. So we lose a little bit of that naive or unbiased nature, but we also get a benefit in terms of increase in calculation speed. So speaking of speed, rigid docking methods usually by far the fastest. Build-up methods a little bit slower. Fully flexible docking slower still. If we allow flexibility of both the protein to some degree and the ligand, that's induced fit, that's even slower. And of course, if we allow full flexibility for both the protein and the ligand, that's molecular dynamics, and that's going to take a lot longer yet. So we have a whole set of posing mechanisms now. Next, we have to score the poses and pick out the good ones over the bad. So we can use our scoring function again for those three problems: ranking molecules by binding affinity, the ranking problem; simply classifying a database into active and inactive, the screening problem; and understanding how active molecules bind into the protein binding site, how they interact with residues in that binding site. That's the posing problem. Three problems for scoring, three basic types of solution, three types of scoring function. First, I'm going to look at force field based on simple Newtonian mechanical force fields. Second, very similar in terms of a linear combination of different energy terms, we call empirical functions. And lastly, a rather different approach based on a statistical analysis of interatomic distances from protein-ligand co-crystal structures. I'm going to look at all three in turn briefly, first with force fields. So here we're using well-established physical models that have been used for decades to predict condensed phase properties for molecules. They use a pretty simple set of functions to look at intramolecular and intermolecular energies, and the overall energies are a linear sum of the individual contributing pieces. We have a lot of advantages with force fields. Decades and decades of use. When we use a force field, we almost literally stand on the shoulders of giants. Workers in quantum mechanics, force field theory, condensed phase matter theory, and so on. A great many contributors. There's a lot of information and knowledge in these force fields. Disadvantages for fundamental perspective. A force field really accounts best for enthalpy. It can be made to produce an entropy estimate, but it's much more challenging. And perhaps peculiarly, for docking, force fields may use a little too much physics. What's that, I hear you say? Well, if we look at the non-bonded term here, there's this what's called the Leonard-Jones or the LJ potential with that term, the 12-6 term. If you plot that based on the distance between the atoms, the optimal contact point is just when the two atoms have their surfaces touching. And as soon as the atoms move any closer, they start to climb this very steep wall, very small changes in distance resulting in very large changes in energy, and that's due to the 12 part of the 12-6 term. So repulsive clashes, bad clashes, it can easily occur in docking, but in nature they're very easily eliminated by tiny adjustments to the protein and the ligand structure. And again, recall most docking programs don't use protein flexibility immediately, so that's not necessarily available. So several approaches have been taken to try to fix this. One is to look at the 12 part potential to make it less steep, make those clashes less penalizing. Then adjustment to say 1 on R to the 9 may be helpful. In other cases, to look at the radius and actually to adjust the radius down from its physical distance to something smaller. Again, these are non-physical terms, but they help the force field be more applicable to the docking problem. One could, of course, refine the pose against the force field and rescore, but that's much more computationally intensive than the other two. So this has led a lot of developers to move away from a full physics-based force field to what's known as an empirical function, which, like a force field, is a linear combination of individual contributions, which are written down by the developer hoping to capture all the key terms. An example scoring function is here. This is the glide scoring function, and you'll see that cleverly the developers here have actually elected to classify hydrogen bonds into 3 different types to improve accuracy. Physically, of course, there's only 1 hydrogen bond, but here, to improve both post-prediction and virtual screening, they've classified them into 3 parts. The weights, the coefficients for these individual contributing terms are arrived at by usually linear, though it could be non-linear fitting, to experimental protein-ligand poses with known binding affinity. So these functions are parameterized against affinity, and therefore we expect them to be better at predicting affinity than predicting poses, because they've seen affinity during training, but not poses. Advantages here is these have an intuitive functional form that's quite easy to understand, and they can be customized, reparameterized for different protein types, and that can be quite productive. Disadvantages include, since you write the function down a priori, did you capture all the key physics? A necessary question with the fitting process is rare interactions aren't going to be accurately handled. The noise in the fitting will be put into the rare interactions, and the frequently occurring interactions will be better handled. So if you have protein ligand complexes that have rare interactions, that's interesting. These functions are going to be difficult to be generalizable. And just purely from the point of view of the experimental data, is it accurate and precise enough to generate reliable and generally applicable coefficients so that your scoring function works for a wide variety of protein ligand types? And this has led a number of developers to move away from this altogether into what are known as knowledge-based functions, where the score for a particular molecule is the sum of the probability of finding each atom in the molecule at a certain distance from atoms in the protein. So we have a distance term here based on a particular pair of atoms, and then a probability of finding the atoms at that distance. And the sum of those contributions is the score for the molecule. These functions are parameterized purely on protein ligand complexes. They don't use any affinity data, so they should be better at predicting poses than they are at predicting affinity. Significant advantage for these is they're unbiased, they're not over-physicsed, they're not predetermined. We simply look at the data in an unbiased way and ask about frequency distributions for interatomic distances. A significant disadvantage is, again, are those interatomic distances sufficiently accurate and precise to give good coefficients in these terms, in the contributions to the overall score? So we have a lot of posing approaches and scoring approaches. A third dimension of complexity, unfortunately, is that for a given particular protein type, docking scores can vary quite dramatically. They're distributed differently. So here I'm looking at two proteins, base 1 and COX-2. COX-2, here in orange, is a deep, hydrophobic, pretty rigid binding site. Here, base 1, open, flexible, formally charged residue in the middle. Same set of molecules, about 30,000 molecules from a public database, have very different score distributions in these two different proteins. Very normal for base 1, skewed with a very wide variance for COX-2. So as you probably already realized, docking scores by their nature are unbounded. They're a sum of atom by atom contributions, so they don't have a defined lower or upper limit, and the nature of the distribution depends on the protein target. So it's very difficult to assign significance. Is this a really interesting score for a molecule? Without doing experiments of this kind, it's hard to know what that is a priori. So now we've got all our pieces in place. We have ways of posing, we have ways of scoring, we understand the differences between scoring and different protein targets. So let's go and actually do some work with docking. And I'm going to first look at the lead optimization problem, finding improvements to molecular structure to improve drug-relevant properties. Now, it looks a little bit like I've accidentally not covered the whole box here. I do this deliberately to illustrate that, as I'll show you in a moment, not all problems of interest in lead optimization are reliably soluble with docking. We'll have to use different approaches. So, speaking of approaches that are difficult, I'm going to start with the hardest problem, using docking to rank protein ligand pairs by the free energy of ligand binding. So to understand this at a physical level, we need to look at a variety of different parts of the process. To end up with the protein ligand bound in solution, we'll need the unbound conformations of the ligand, the unbound conformation of the protein, the two of those desolvated, then in a vacuum, and then finally together. Docking, as you probably realized, actually only deals with the bottom right corner, taking a conformation of the ligand, placing it in the protein, and scoring that interaction. So docking doesn't know about the unbound state of the protein or the ligand, and that's going to make it difficult from a fundamental point of view to be reliably accurate for binding energy prediction. Also, as I mentioned, most scoring functions focus almost entirely on enthalpy. As you see here, we're looking at plotting enthalpy from experiment against free energy from experiment, from isothermal titration calorimetry. Enthalpy doesn't predict free energy. And I'm sure you can see what's coming. We can't also use entropy to predict free energy. We need both at the same time to accurately assess binding affinity. So from a fundamental physical point of view, docking should struggle with reliable predictions of affinity. Looking at prospective experiments, this is from something called the D3R, or the Drug Design Data Challenge. So these are prospective experiments where the contributors don't know the correct answers until after they've submitted their best predictions. Here, for a particular kinase, this is the Janus kinase 2, I have about 18 predictions. The measure of match between the prediction and experiment is measured with something called the Kendall's tau, higher, more positive being better. A different protein, VEGFR2, more submissions. Rather more difficult problem. As you can see here, the Kendall's tau range only goes up to about 0.5, the best tool here about 0.45 versus about 0.7 for JAK2. Now, if we look at the Kendall's tau and say, well, generously, a Kendall's tau of 0.3 or more, again, measuring the association between the prediction and the experimental ranking, more than 0.3 is going to be a useful model, possibly. For JAK2, only 3 out of 18 submissions have a tau outside the error of greater than 0.3. And unfortunately, within error, 14 out of 18 show no correlation, tau of 0, or an anti-correlation, tau less than 0. For VEGFR2, a little different. Only 1 out of 33 has a tau greater than 0.3, but only 4 out of 33 have a tau less than or equal to 0 outside the error. But both of these prospective experiments, I think, say the same thing. Affinity prediction in a prospective setting is very difficult. So generally speaking, docking is not a reliable approach. There are certainly cases where it works very well, but it's very difficult to generalize. So for the rest of this, I'm going to focus on tasks I think docking will be more appropriate for. Firstly, in the lead optimization area, to understand how active molecules bind to and interact with the protein binding site. So to test this, we're going to need several things. Most importantly is a good quality structural model. If you want high precision post-prediction, you need a high precision atomic coordinate model. We can use that model to do 2 types of post-prediction experiment, what's known as self-docking, where we take the molecule out and put it straight back into its own binding site. Pretty easy problem to solve. If you have this, you may have something, but if you don't have this, you don't have anything. Second, the much more difficult task, the prospective task, a whole set of different molecules into a single protein binding site, and that's called cross-docking. Lots of different ways of measuring success in post-prediction in either of these environments. Lots of different metrics here based on deviation from the coordinates of the atoms, things like RMSD, used all the time, but as I'll point out in these papers, a terrible measure apart from that, based on other aspects of the atomic coordinate model. You should already be thinking, well, isn't the atomic coordinate model just a model? And shouldn't I compare to experiment? And you'd, of course, be exactly right. You should compare your predictions to experimental data. The atomic coordinates are just models, so you should compare to the original data. So for electron density, that would be something like the real space correlation coefficient, RSCC, or the real space R factor, RSR. It's quite rarely done, but if we were to predict log P or solubility, you would compare directly to the experimental measure, not to some abstracted measure. However, the great majority of docking papers will return something like this. Some deviation between the predicted and the experimental pose, here they're using RMSD, and it's a generally considered community standard. If your RMS is less than 2, you've done a successful job with posing. And there are a great many papers out there looking at comparing tools for post-prediction success. I just picked on one here. Looking at a variety of different tools, using different aggregate measures for overall performance, median, mean, some estimate of the variance, and most importantly, an estimate of what's known as the confidence interval. They also delve more deeply in this paper into comparing different methods. In the upper triangle of this table is the correlation coefficient measured using the Pearson coefficient between results from different tools. So the lower this number, the less closely the two tools correlate, the more they behave differently on different systems. In the bottom left, some statistical significance estimators of those correlation coefficients. If you'd like to know more about confidence intervals and decision-making generally in molecular modeling, please join me for Essentials Part 4. Looking at another way of comparing different methods, we could ask what's the probability that a particular tool is going to outperform another one. So here's a whole set of tools compared again for post-prediction. And as you can see, the great majority of numbers here range between about 50% down up to about 75 and down to the 40s and 30s. So in short, there is no high statistically certain difference between any of the tools in this set, and I'll come back to that in the next section of the talk. So again, this is retrospective experimentation. For prospective comparison, again, I'll use the D3R, the Drug Data Design Challenge. This is docking about 24 molecules into cathepsin S. The mean RMSD here, and as you can see, all the tools have a mean RMSD greater than 2, so not particularly great success here, but at 3 angstrom there may be some useful information. Here, interestingly, of the top 5 tools by accuracy, all tools use some engagement, some information from the ligand, be it for conformation selection or deeply embedded in the pose algorithm. But in all cases, ligand information here is giving better results, as I indicated it would in the earlier section. So that's the lead optimization part. Affinity prediction, very challenging. Pose prediction, much more accessible. I'm going to conclude by looking at an earlier part of the lead discovery process, and that's compound screening. And again, I leave this for last because this is by far the most popular use of docking tools over the decades. Virtual screening, ranking a database so the active molecules go to the top. And again, I mentioned this in a little bit of detail in part 1, looking at ligand-based methods, 2D and 3D. And again, we're going to be looking at structure-based design, particularly docking here. And again, to remind you, the key attraction for docking is the novelty of the molecules. And I'll show you some examples of that through this section. So here we're looking at that screening problem to discriminate binders from non-binders. And I'm going to look here at molecules to be screened coming from what I'm going to term pre-generated collections, sets of molecules that have been defined for you, a little bit like defining an area for a mine. So these pre-generated collections I'm going to divide into 2 parts, 2 types. Annotated data sets, things like PubChem, where there's data of at least one biological assay for every single molecule in the data set. And these data sets have some significant advantages. You can generate targeted subsets, all the molecules that are active against a GPCR or a kinase, for example. The libraries are relatively small, the largest ones here being only about 100 million molecules. Disadvantages include there's no necessary route to obtaining the molecule. You're probably going to have to synthesize it yourself. So a lot of at-risk chemistry. There's nowhere to necessarily buy it from. And from the intellectual property perspective, the molecules themselves have no novelty. Everyone is able to search them. They're all publicly accessible information. So the novelty only comes from the use of the molecule on your particular target. The alternative are what I'll call unannotated collections, no biological data attached. And here, these usually come from vendors, companies like Iridium, Liverpool Cryo-EM, and Wuxi. These can be gigantic collections, multi-billion, multi-hundred billion in some cases, and these provide a lot of interesting advantages. The size of the library, as I'll show in a moment, is usually considered an advantage, but of course there is the large amount of computational cost that comes with that. And the business model of these vendors is very simple. Molecules for money. You exchange money for the molecules. They have very good ability to predict success rate and delivery times. These are very efficient ways to obtain molecules. But again, a significant downside is that for the most part, there's no novelty in the molecules in these databases. Anyone can access the structures. Anyone can search them. The novelty comes in the application of your particular target. So why are these vendors looking to make these utterly enormous libraries, which have been increasing in size all the time? Well, it's because these libraries give you better answers. Here I'm looking at a database of about 90 million molecules from what's known as the enamine diverse set. And as we go left across the plot, we're taking increasingly small subsets down to only 10,000 out of the original 87 million. And what we're looking at here are the scores for the top 50 molecules in each of those sets. And as you can see, they increase monotonically with every increase in library size. So if you believe the scoring functions are worth something, and they certainly are, these larger molecules have better hits. So there's going to get better hits, possibly some interesting structural diversity. Unfortunately, of course, the calculation time will scale more or less linearly with the number of molecules in the database. So thinking of virtual screening now, how are we going to assess accuracy? I'm just going to look at a single metric, which is called the area under the curve for the receiver operator characteristic, the AUC. It's false positive rate on X and true positive rate on Y. This measures something called discrimination, the probability that given an actual active and an actual inactive, the active gets a better score than the inactive. So random performance, 50-50, 0.5, perfect performance, 1.0, 100% ability to rank true actives over true inactives. So here I'm going to show some retrospective virtual screening performance measured using AUC here on the y-axis, 0.5 random, 1 is perfect. A wide variety of tools here. We've got stochastic, deterministic, rigid, flexible, build-up, ligand-assisted, the whole set. As you can see, there are some numerical differences in the performance, but if we look for statistically significant differences, meaningful differences over time, those are indicated by the green squares. Only a very, very few pairs of tools show statistically significant difference. And if you'd like to learn more about this and how you can apply it, deciding between different methods in CompChem, please join me for Essentials 4. So now going away from retrospective performance into prospective performance, this again is from the D3R experiment. Looking at AUC, 0.8 here, this is virtual screening into the farnesoid X receptor. So really quite good, about an 80+% discrimination, very solid virtual screening performance. But the same set of molecules, when we plot their experimental affinity on X versus the predicted on Y, there's a very poor correlation here, tau is 0.4. So for the screening problem, this tool works very well, but for the ranking problem, it's not particularly good. So again, good performance for virtual screening, not so good for predicting binding affinity. So now I'd like to move to an experiment that we actually did here at OpenEye a few years ago for prospective virtual screening against a target protein called heat shock protein 90, HSP90. There's the binding site with a small co-crystallized ligand. So in this experiment, we took about 1.4 billion molecules from the enamine collection, added a number of known actives from a public database as a computational sanity check, docked and scored all 1.43 billion of 47, selected some interesting scoring molecules from the top of the list, bought them and sent them to a CRO for assay. One of our very interesting hits is shown here in its docking pose. There's the hit molecule in gray. The crystal molecule here is in magenta. So they make similar but not identical interactions to the left of the pocket and interactions on the right only with the new molecule. The existing known ligand, quite a weak binder, only about 50 micromolar. This molecule, however, has a substantially higher potency, IC50 about 10-fold better. So here we see the promise of docking fulfilled. The structure of the new ligand completely different in every respect to the existing ligand, and again, turns out to be substantially more potent. If we compare this ligand to one of the top ranked hits from our three-dimensional similarity search engine, FastRox, which we did exactly the same experiment on with the same molecules, this is one of the top ranked hits, different in chemical structure to the query, to the original ligand, but rather similar in size. Distribution of interactions in the binding pocket. So again, docking has found something entirely novel that also turns out to have rather interesting affinity properties. So let's have a look at what we should and shouldn't do with these virtual screening experiments. The second thing I'd like you to take away from today's presentation is you're always going to get the top N molecules. Don't just take the top N, go off and buy them, synthesize them and test them. What you do want to do is critically think about what you'd like out of your data set and try to extract the maximum value you can. You spend a lot of time and effort preparing the database and searching it. A little more time and effort into the analysis will pay dividends. So you can look for more value in terms of more information, more structural diversity, either in 2D or in 3D from your hits. You can look for the most interestingly scoring molecules. Again, the hope in any virtual screening technique is that the active molecules here in gold have a different distribution to the inactive molecules here in blue. So to find that difference, to look for significantly interesting scoring molecules. You'll always have top scoring molecules, but are they interestingly scoring? And lastly, increase the confidence you have in the decisions that you make. You're making at-risk decisions for money, time, resources. Make sure you maximize your confidence. So to illustrate that, I'm going to show, I think, a nice example of how ligand and structure-based methods are complementary. This is some work from Sanofi Aventis a few years ago, looking at virtual screening into 4 targets, where they used 3 different virtual screening methods. Docking with glide, 3-dimensional similarity with rocks, and 2-dimensional similarity with feeder trees. For each of the circles in the Venn diagram is the number of unique active hits found in the top 50. So all the tools do very well here. But look at the overlaps. These are the overlaps between any pair, and in the middle in white is the overlap for all 3. And as you see, any overlap produces more active molecules than any individual contributor. They find different active molecules at the top of the list because they look at scoring molecules in completely different ways. So what I'm going to suggest is to leverage this kind of knowledge, this kind of different approach, and instead of trying to find the correct tool for your particular problem, your particular protein, your particular molecules by doing retrospective tests, which is possible, to pick methods that seem appropriate for screening the molecules you're interested in, and then combine them using consensus or data fusion methods. I'll just illustrate how that might work here. This is virtual screening success. Higher is better. The performance for 6 individual techniques, 2D and 3D ligand-based and structure-based, on the left, and then on the right are 3 approaches to combining the results from these individual methods. And as you can see, the mean and median performance for these consensus methods can be substantially better than any individual piece. So again, intelligent data fusion makes a big difference. So, what did we look at today? We looked at screening molecules, protein conformation, and we looked at docking to solve 3 related problems. Ranking so that the active molecules go to the top, the classification problem, and that works well with docking. And again, it's traditional use over time. Pose prediction, understanding how an active molecule fits into the binding site, works quite well. Free energy of binding prediction, much more challenging for docking, and if you're interested in computational approaches to that, particularly using molecular dynamics, please join me for Essentials Part 3, which will be on affinity prediction in lead-opt. So, in summary, we've looked at a lot of things today. We've looked at determining structural model quality, wherever they come from. We've looked at pose prediction methods, rigid, flexible, stochastic, deterministic, etc. We've looked at scoring functions, force fields, empirical functions, knowledge-based functions. We've looked at pose prediction, retrospectively and prospectively. We've looked again at lead discovery in a prospective context, and we've looked at the importance of consensus or data fusion methods. So if you'd like to pursue some of these ideas further, please get in touch to see if some of our software may be helpful. If you're in academia, we offer our entire software suite for free to qualified academics, so please get in touch. If you'd like to join me for Essentials Part 3 on lead optimization and affinity prediction in lead-opt by MD, please join me on the 12th of November. Otherwise, until then, goodbye. Welcome everyone to the Q&A. I hope you enjoyed the recorded talk. So I'm going to handle some questions that have come in over the course of the talk. So I'm going to start with one that just came in on using rule of 5 criteria for filtering hits from virtual screening. If you want some more details on this, everybody, please go and have a look at Essentials Part 1. But I think the short answer is it's a great idea to filter your database before you go into computationally more expensive methods like docking. So filter before whatever rules you'd like to apply. Traditional drug likeness filters, things like the rule of 5, are aiming to try to find molecules that are soluble, permeable, have probably good bioavailability properties. Filtering on traditional rule of 5 to find those kinds of properties is quite difficult. And again, I expand more on this in the Essentials Part 1, but basically simple properties used in rule of 5, things like log P and molecular weight, don't necessarily reliably identify molecules that are going to be soluble and permeable. So filtering before docking, a good idea, but be careful the sorts of properties that you filter on, so you're making sure that the molecules that survive the filtering get the properties that you need. Okay. So a question came in on scoring functions. So I talked a lot today about quite simple, fast scoring functions, force fields, empirical, knowledge-based, and so on. So a question came in about using higher levels of theory, quantum mechanics for scoring. There's been quite a lot of work in this, and I didn't have time really to go into it today. The promise with higher levels of theory is, of course, higher accuracy. And there are some cases where that can be true, but it's not always clear that higher levels of theory are always more accurate and more functional, and you'll pay a huge penalty in time. Having said that, there's some very interesting recent work come out from Pavel Hobz's lab on a method he calls SQM 2.0 that was published quite recently, looking to assess binding affinity from docked poses using semi-empirical methods to compute entropy and enthalpy. And that appears to work quite well. So I think there is some promise there to use higher levels of theory, though not necessarily as high as DFT, but certainly semi-empirical methods appear to offer a nice increase in accuracy for not an enormous increase in scoring time. Okay, let's see.
Oh, well, I'll take another one on a related issue, using machine learning. So I deliberately didn't look at machine learning in scoring or docking today. That's a huge field that's growing very, very quickly. And unfortunately, I think anything I'd say today about machine learning in docking or machine learning for scoring would be out of date by the end of the week. So yes, there's a lot of potential for using machine learning for scoring. I think that's worked quite well. I think there's a lot of opportunities to think about how to use the data sets correctly. Again, something I touched on. And machine learning functions like any other function. If you train it on poor data, it's going to work poorly. If you train it on good data, you have an opportunity for it to work well. Question here on using AlphaFold. Again, I deliberately didn't look at theoretical predictions. It's a really interesting new area. AlphaFold proving to be very successful for predicting protein structure. There's been some interesting work recently from Brian Choquette's lab on this exact question, using AlphaFold to predict structures versus using experimental structures, and apparently they work reasonably well for virtual screening. Perhaps not for pose prediction, and that's perhaps not unsurprising that the details of atomic arrangement in binding site have a strong effect on posing. AlphaFold is probably not going to be particularly great at that, but in terms of arranging the binding site generally, so that molecules can pick up the correct interactions, I think it works quite well. So yeah, I think AlphaFold structures for virtual screening, the experiments done so far seem to say yes. For AlphaFold in the pose prediction problem, I think the structures need to be more closely refined before we could use them effectively. Let's see. Oh, yeah. So in terms of testing, I showed some results for prospective and retrospective work, and somebody pointed out that the retrospective results look usually a lot better than the prospective, particularly for pose prediction, and that's true. Why is that? Well, there are a couple of reasons. Retrospective tests tend to get run multiple times, and so there's an opportunity for what you might call data leakage to occur. There's a possibility that one can optimize your tool's performance, usually with it unintentionally, so that it performs well on tests that you know the answer for. And this is a big problem in any parameterization or model building. It's not just docking. Machine learning pointed out quite a lot recently that if you don't build your model correctly, you are going to have problems in generalizing, making good predictions outside your training set. And the same goes for scoring functions. So I think we've seen that prospective performance is also difficult because the proteins that are used in these prospective sets tend not to be the same proteins that people use all the time. They tend to be interesting proteins on the cutting edge of a certain project, for example, cathepsin S, not one of the post-prediction data sets, is not a very well-used protein in evaluating docking programs. So I think it's not surprising that the results don't always generalize. I would also point out that when we're thinking about doing very, very important generalizations, so for example, in clinical trials, where we're actually using humans to test the particular treatment, we approach testing in a rather different way. And I think something I would like to see happen in this area is for us to take a more clinical, so to speak, more sort of medicinal driven, so thinking about our cases more carefully, thinking about construction of our data sets more carefully, thinking about accuracy predictions more carefully, so that when we do make the predictions, we end up with better answers. Okay, one more question. So I talked a lot about scale today, and somebody asked me, so which docking programs work best at very large scale? There's not an enormous amount of data on this, but I think what we've found, certainly internally at OpenEye, when I showed you some very large scale work we've done earlier, that the rigid docking approaches tend to work well. There's some work from, again, Brian Choquette's lab and others, where things like Doc3 and Doc6, some build-up methods, or anchor and grow type approaches, can also scale quite well. Too much flexibility in docking tends to increase the computation time, so it becomes prohibitively expensive to dock 1 billion, 3 billion, 5 billion molecules. It takes too much time. So I think either the rigid methods or anchor and grow methods seem at the moment best suitable for very large scale. So I think at the moment we've come to the end of all the questions. So Thank you all again for attending the Q&A. I hope you got some utility out of both this and the presentation itself. And I look forward to seeing all of you the next time for Essentials Part 3 on relative binding for energy prediction in lead optimization. So until then, thank you. Goodbye.